Every living being on this planet can be classified into two main types. First one is cellular organisms and second one is non-cellular organisms. These non-cellular organisms consists of viruses and viroids, whereas rest of diversity is included in the cellular organisms. Cellular organisms can be further classified on the basis whether there is nucleus present inside cell or not. If nucleus is absent, we call them prokaryotes, and if nucleus is present, we call them eukaryotes. The word pro means before and karyon means nucleus. So prokaryotes do not have nucleus, whereas the term eu means true and karyon means nucleus. So eukaryotes are having a true nucleus. Let's talk about prokaryotes. These prokaryotes can be further classified into archaea as well as eubacteria. Eubacteria are the classical bacteria we know of, whereas archaea consists of some specialized type of bacteria which are found in some extreme habitats. Eukaryotes can be classified further on the basis of number of cells present. If there are unicellular type of organisms, we place them under protists, and these are called unicellular eukaryotes. Whereas if they show multicellularity, we call them multicellular eukaryotes. That is as simple as that. Non-cellular organisms we already talked about. It includes viruses and viroids. Let's talk about five kingdom system of classification. It was given by Robert Whittaker in 1969. The first basis is cellular organization. We talked about earlier that how cellular organisms can be classified into prokaryotes and eukaryotes. This is that. And another basis is cellularity, which determines whether they are unicellular or multicellular eukaryotes. As you can see, there are five kingdoms which are Monera, Protista, Fungi, Plantae, Animalia. The Monera is exclusive for prokaryotes and they are unicellular in nature, whereas protists, which are eukaryotes but still unicellular, and rest of eukaryotes, which include fungi, Plantae, and Animalia, are multicellular eukaryotes. And in nutrition, we study the mode of nutrition and mode of uptake of that specific type of nutrition. Let's talk about mode of nutrition. So mode of nutrition at cellular level can be classified into three main criteria. First one is what is the source of carbon, which is responsible for formation of the substrate like carbohydrate. Second one is what is the source of energy which was required to make that specific carbohydrate or any another substrate. And third one is what is the source of electron which was involved in that specific metabolic pathway. So these are the three bases on the basis of which the mode of nutrition can be classified, and we call that cellular basis of classification of the mode of nutrition. Let's talk about each of these criteria in details. If the source of carbon is inorganic carbon, we call those organisms autotrophs. If the source of carbon is organic carbon, we call those organisms heterotrophs. Now let's talk about source of energy. If the source of energy is light, we call them phototrophs. If the source of energy is some kind of chemical compound like sulfur dioxide, we call them chemotrophs. Let's talk about source of electron. If the source of electron is any inorganic compound, we call such organisms lithotrophs. If the source of carbon is some kind of organic compound, we call those organisms organotrophs. Let's test this with some examples. First example is Homo sapiens. If we talk about the source of carbon, it is organic carbon. So we consider Homo sapiens as heterotrophs, which is specified as heterotrophs. If we talk about source of energy, the source of energy is not light. It is some kind of chemical compound. So we are chemotroph as well. And if we talk about source of electron, so we take electron from organic compounds. So human beings are chemo-organo-heterotrophs. Let's talk about another examples, which are green plants. Now, if we talk about source of carbon, the green plants utilize inorganic carbon which they absorb through their roots. So plants are autotrophs. If we talk about source of energy, the source of energy is directly light because of the photosynthesis. So we call plants phototrophs. If we talk about the source of electrons, the source of electron is also inorganic compounds which are absorbed through their roots. So we can say that green plants are photolithoautotrophs. Similarly, we can classify fungi into chemoorganoheterotrophs. We can classify cyanobacteria because cyanobacteria are photosynthetic. We can classify cyanobacteria into photolitho and autotrophs. We can classify nitrifying bacteria which are responsible in the nitrification process. We can classify them as chemolithoautotrophs. Now let's come to some specific terms regarding the nutrition uptakes. We already classified and defined the terms like autotrophs and heterotrophs because these autotrophs use inorganic carbon, primarily carbon dioxide, as their principal carbon source and synthesize some kind of carbohydrate. So these are the autotrophs, which are also called sometimes producers in ecological terms. In case of heterotrophs, heterotrophs use preformed organic carbon, and this preformed organic carbon is usually from this autotroph or another heterotroph. So autotrophs use inorganic carbon and heterotroph use organic carbon as their primary carbon source. We already talked about autotrophs and heterotrophs in previous slides. Let's come to consumers. Consumers are the heterotrophs that ingest biomass. Now the term ingestion is important here. Now they ingest biomass, but it is not to be considered whether this biomass is from living or dead organism. Both can be considered under the term consumers. Now let's come to more specific terms. If some heterotroph is absorbing or ingesting nutrients directly from living or directly from living hosts and ultimately harming that host, we call such organisms parasites. If some heterotrophic organism is ingesting only dead biomass, we can call them detritivores. And if some kind of heterotroph extracellularly digests and then absorbs dead biomass, just like fungi, we call them saprotrophs. Under saprotrophs, there is one more category which is decomposers, and decomposers are specific saprotrophs that carry decomposition to the level of elemental mineralization. Let's come to three domain system of classification. Three domain system was given by Calbus in 1990, and the major basis of the classification was the evolutionary relatedness and molecular data. So it was based on molecular phylogenetics as well as comparative genomics. They were comparing some specific oligonucleotide signature sequences of 16S ribosomal RNA, and those having same oligonucleotide sequences were placed in the same category. That's how this three domain system of classification was initially given. So they were comparing the ribosomal RNAs as well as core molecular machinery, which includes the mode of replication, how transcription and translation were occurring in these organisms. So despite the mode of nutrition, whether they were autotrophs, heterotrophs, or whatever, these modes were not considered. And ultimately the metabolic diversity was existing within the domains, not between them, which means that we were only focusing on the evolutionary relatedness between the organisms despite having diversity in case of the nutrition or mode of uptake of nutrition. So there were three domains: bacteria, archaea, eukarya. Eukarya includes all the eukaryotes, and eubacteria and archaea includes the prokaryotic organisms, which if we compare them directly to the five kingdom system of classification. We have last universal common ancestor, which is giving rise to one group of bacteria and some another type of common ancestor, which will eventually give rise to archaea as well as eukarya. So this means that archaea is more related with eukarya as compared to bacteria. And if we talk about specific features and their relatedness, the cellular organization of archaea is related to eubacteria more than any kind of eukaryotes. Whereas if we talk about molecular machinery, metabolic processes, and all the genetic processes like replication, transcription, translation, this is more close with eukarya. Let's come back to non-cellular organisms, and we are starting with virus. Now virus has chemical nature of nucleoprotein. By nucleoprotein, I mean two components. First one is nucleic acid, another one is protein. Nucleic acid acts as genetic material and it may be either DNA or RNA, but never the both. And these nucleic acids may exist as single stranded form or double stranded form. It may be linear or circular. If we talk about proteins, proteins will form capsid of the virus. According to capsid, the shape of the virus will be decided. This capsid can exist in various shapes like icosahedral, which looks like some spherical form, as you can see right here in this diagram. There is helical form as well, which looks like the helix. There is also complex shape as well. Now there are some surface proteins which are responsible for recognition by the host cell, and that's how the virus will invade the host cell or host tissue. Now the host of the virus may be different kind of organism, but they have to be cellular organism only, and it may be residing inside prokaryotes or eukaryotes. So we can also say that the viruses are obligate intracellular parasites. Bacteriophages will be specific for the bacteria. They will reside only inside the bacteria. Plant viruses attack only plants, and animal viruses will infect only animals. Another thing to remember is that viruses are metabolically inert outside the host cells, which means that viruses lack any metabolic pathways, and for their reproduction or for their division they are entirely dependent on the host cells. The viruses have been evolved by the natural selection, which is quite obvious. Viruses also mutate and they also show variation patterns, but they lack autonomous life processes. That's why they will need specific cellular organisms to reside in. The second non-cellular organism we will talk about is viroids. Now these viroids or viroids are chemically nucleic acids. How are they different from viruses? Viruses were nucleoproteins, which means nucleic acids as well as proteins, whereas viroids are only nucleic acids. And nucleic acids very specific, like they are small circular single stranded ribonucleic acids. They will form a rod like secondary structure, and that is why they are chemically stable but biologically inert RNA, we can say. Another important characteristic to remember in case of viroids is they do not encode for proteins. And if they do not encode for proteins, they will lack their own enzymatic activity, and all of their biological processes will be dependent on the host proteins, or we can say host enzymes. They will replicate using host RNA polymerases. All of their biological activity is encoded in the RNA structure itself, but not proteins. For proteins, they will utilize the host proteins as well as host enzymes. If we talk about the replication of viroids, they replicate autonomously inside host cells using host enzymes, which we already talked about, that they are host RNA polymerases. The mode of replication is rolling circle mode of replication, and this replication of viroid takes place inside nucleus or plastids, and it is specifically RNA directed RNA synthesis using DNA dependent RNA polymerases. They are the smallest known infectious agents, and their host is specifically the plants. There is not a single animal, fungal or bacterial host known for the viroids. For now, they are exclusively for plants. We can also say that they are obligate intracellular parasites in plant cells. What is the actual mechanism of infection of viroids? So, viroids infect only plant cells, and they do so by RNA mediated interference with host gene expression. They do RNA silencing, they may alter the transcription, they may disrupt the host regulatory RNAs. So we can say that the pathogenicity is regulatory but not strictly biochemical. This is all about viroids. Let's move on to next topic,